Okay, so my thesis title is Effects of Encapsulation on the Viability of Probiotic Lactoacidophilus in Yogurt. Sorry. So for the introduction, good. So yogurt is fundamentally fermented milk derived from bovines or cows. So ito yung health benefits it's known for, attributed mainly sa lab cultures. So ang pinaka-popular benefit is yung improving intestinal tract health. So it's important na yung human gut microflora natin ay ma-maintain in a viable population, and we do that by consuming fermented products like yogurt. So traditionally, two starter cultures are required. We have standards for that, and yun yung Streptococcus thermophilus and Lactobacillus bulgaricus. But hindi sila deemed as probiotic dahil hindi sila nagso-survive sa ating sa gastric conditions or sa ating tiyan. With that, probiotics term was originally intended to describe microorganisms that contribute health benefits to the host. So that's why nag-include ang yogurt manufacturers ng probiotic strain, and only then yung yogurt naka-classify as probiotic yogurt. So of course, we have a requirement pagdating sa viability or count, which is 10 raised to 6 CFU per grams for the whole shelf life duration. And this is because the higher yung viability, the more din yung health benefits that the host will receive. So, but the problem is, however, there are numerous literature citing the non-viability of probiotic products due to the following factors. So, Lactobacillus acidophilus is the most studied and utilized probiotic strain. So, ito yung ating gagamitin sa ating study. So, this is the definition of microencapsulation, which is the method that we will employ to maintain the viability of probiotics in the yogurt. So, and by the way, on the right is the encapsulation by extrusion method. So we have 3 variations ng microencapsulation to be applied, and 2 of them ay encapsulated yung probiotics, and sa isa, yun yung magsisilbi nating control, wherein non-encapsulated ang probiotics. So statement of the problem. So the study aims to determine the effects of encapsulation by answering the following inquiries. So what are the effects in terms of viability, pH, titratable acidity, to TSS, and viscosity? Are there marked differences among the variants in terms of viability, pH, TA, TSS, and viscosity? And if those differences are significant enough, including the sensory parameters as well. Okay, for the methodology, so here are the ingredients for the yogurt. So we used a Nestle Greek yogurt as the yogurt base. And ito yung components ng encapsulation, and I have to mention that we obtain probiotic culture in active form from the Biotech Institute. So ito yung components for enumerating yogurt, and of course we have to use a differential media para Lactobacillus acidophilus lang talaga ang tutubo. And from a study guide, I've seen sodium acide used at the rate of 0.02%. So this is the process flow for the whole study. So single phase lang siya. So for the first step, purchasing the cultures, as I've mentioned, the probiotic culture was obtained in active form. So upon receiving, it was subcultured, pinarami, and then inoculated in MRS broth. So the inoculates are then incubated at 37 degrees Celsius for 24 hours at anaerobic conditions. So in preparing the yogurt, of course, minix muna yung yogurt ingredients. However, for the control variant, and by the way, inuna mo nang ginawa ang yogurt, and the yogurts are dispensed before pinropagate yung I mean, yeah, so I'm sorry for that. Okay, so for the control variant, so aseptically inoculated twice na lang from a reserved subculture at the rate of 1%. And the yogurt mixes are then incubated at 45 degrees for 12 hours and then dinispense sa respective lots. So, in the study I've seen, actually 80 ml ang na-harvest na probiotic biomass. But then, due to the limited resources, we were only able to harvest 40 ml ng probiotic biomass upon low-speed centrifugation. And after siyang i-harvest, it was washed twice in saline solution, in sterile saline solution under the same centrifugation conditions. So, for the wall material mixture, dalawang varieties ang pinrepare. Each is 60 ml. And of course, half ng harvested culture or 20 ml each was mixed to each wall material mixture. So for the encapsulation by extrusion, so yung mixture, dinrip siya using a syringe above a hardening solution which is 0.1 m of calcium chloride. And of course, yung water is cold according to as advised by and then upon dripping, nagfo-form ng microcapsules instantly. So yung after the beads are then collected by decanting the hardening solution and evenly dispensed sa pre-dispensed na yogurt lots. So in enumerating the culture, sinosolubilize muna natin ang beads to release the cells inside. So we do it by suspending the yogurt sample with microcapsules to a phosphate buffer solution and sinishake siya. So it's supposed to be 10 minutes, but due to time constraints, 1 minute lang. So for enumerating the L-acidopilus, we employ pour plate method. And of course, as I've mentioned, infused with 0.02% sodium acide ang ating MRS agar for differentiation. So incubated at 37 degrees Celsius for 48 hours dahil medyo mabagal siya tumubo in anaerobic conditions. So after, the plates with colony counts in the range of 30 to 300 are then selected. And as for the control variant, same procedures except with the solubilization step kasi wala naman siyang capsules. So of course, in pH determination, we use a pH meter. And for the TSS determination, we use a refractometer. All are calibrated, of course. So for the titratable acidity, so I followed a method in which 10 grams ng yogurt sample ay dinidilute sa 30 ml of distilled water and titrated until turned to a faint pink color. So the volume of titrant was used to compute the percent lactic acid. So for the measurement of viscosity, so 200 ml ng yogurt ang na-estimate ko na ginagamit and it was transferred to a leveled Bostwick consistometer, then evened off, of course. The distance traveled in centimeters after 30 seconds is the one being recorded. So, of course, one trial lang because konti lang yung yogurt natin. So, for the sensory evaluation, each day of analysis, 15 untrained professionals were invited, and they score using 9-point hedonic rating scale score sheets. So for the statistical treatment, we use ANOVA to test for significant differences and line graphs as well. For the results and discussion, so this one is for the comparison ng viabilities ng probiotics, and it's obvious na lahat sila may decreasing patterns. And yung control yogurt natin, by the 7th day, hindi na siya viable. But yung ating variants with encapsulated probiotics ay nag-remain na viable. So, ang viable count natin should be, as I've said, 10 raised to 6 CFU per grams or 6 in log. So, statistically, yun nga, there are significant differences. and such that encapsulation with alginate and gelatin significantly enhance the viability of probiotics in yogurt. However, according to statistics as well, encapsulation with alginate plus starch alone have no significant effect sa yogurt. So, gelatin lang yung may significant effect sa viability. So, for the pH naman ng yogurt, so as you can see, increasing siya at first, but then nag-decreasing na siya by the third and fourth days para dun sa encapsulated probiotics variants. But yung pH ng ating control variant ay nag-maintain at 4.8 to at the ranges of 4.8 to 5 for the 7 days ng study. So with that, masasabi natin na no yogurt variant ang nag-meet ng standard pH range. And of course, there are also significant differences among the pH levels. And it's such that the variants with encapsulated probiotics have generally higher pH levels than the variants with non-encapsulated probiotics. So the factors to consider is that the encapsulated probiotics have limited metabolic activities kasi nga they are entrapped, and it takes time for lactic acid produced para to permeate or to get out of the capsule. So ito yung tinatawag nating slower post-acidification rates. So for the titratable acidity or percent lactic acid ng yogurt, as we can see, all of them have increasing patterns. So the minimum TA level is 0.6% and the maximum TA value is 0.9%. So yung variants with encapsulated probiotics generally did not meet the standards. Yung control variant has met but exceeded the max value by the 7th day, which kasi is 1.03%. So statistically, wala namang significant differences. So walang effect ang encapsulation sa titratable acidity. But I believe na due to extrapolation, baka may significant differences that can be observed if longer time yung observation. So for the TSS, as expressed in bricks, so as we can see, of course, obviously may significant differences such that mas mataas ang bricks level ng control variant sa variants ng may encapsulated probiotics. So ang standard milk fat content should be less than 15%. So yung ating EASG and EAS variants both met the standards but hindi yung ating control variant. Iba naman this time. So statistically, there are significant differences, as I've mentioned, and the high TSS is due to the lower probiotic viability in yogurt. And by the way, a high TSS content is related to a lower probiotic viability in yogurt due to slower conversions of sugars into lactic acid. So inversely proportional sila. So I've found this is the same finding with Tan et al. But in this study, it can be seen that yung pH levels and TSS contents ng yogurts ay inversely proportional instead. I mean, for the yogurt with encapsulated probiotics are inversely proportional instead. And I think it's because yung variants, it might have slower post-acidification rates, but at least yung pH is still gradually decreasing over time. So this for reference. So as we can see, yung pH ay decreasing. As we can see, yung pH levels ng variants with encapsulated probiotics, they are increasing over time. Although hindi sila ganun kababa ng pH level sa control variant. So, however, yung control variant, mababa nga siya, pero hindi naman siya bumababa pa. So with that, I can say na siguro, ayun nga, longer observation time, mas makikita yung pattern na the lower the pH, the lower the TSS then, kasi they should be directly proportional. Okay, going back. So yun nga, such phenomenon can explain why the control yogurt have high TSS despite having mainly lower pH and probiotic viability. So for the viscosity, so as we can see, lahat sila ay increasing, and it's saddening na lahat sila ay, of course, did not meet the standards, obviously. 24 is the maximum level actually. So statistically, of course, wala ding significant differences or walang effect ang encapsulation sa viscosity ng yogurt. However, if you can compare yung encapsulated probiotics variants, they have higher viscosities, and it could be attributed sa thickening properties ng wall materials used sa encapsulation. For example, the gelatin, the sodium alginate. So other factors to consider is that there's increase in syneresis or yung pagbi-breakdown ng lactose content ng milk, and medyo nag-contribute din or factor din yung manner of dispensing, because I've noticed na medyo buo pa yung form ng curd ng yogurt before i-dispense, but upon dispensing to smaller containers, nawawala na siya ng form. So factor din yun kung bakit ganun siya kalabnaw or yun na walang form. So for the sensory evaluation scores, of course, summarize na lang to. So we can see na yung color, aroma, flavor, and general acceptability, merong significant differences yung means ng variants. And if we're gonna look at the superscripts, it's always the control yogurt na naiiba or significantly different. So for the color or appearance, we can see na medyo, yun nga, pinaka-favored yung control variant. So, yun nga, it's the only one acceptable based sa mean score, and yung encapsulated with gelatin is the least likely acceptable. So, statistically, there are significant differences as seen here. And based on feedback, so they said yung yogurt variants with encapsulated probiotics have slightly tinged white color compared sa color ng control variant. So it can be attributed din sa colors ng wall materials. So yun nga, off-white for alginate, yellowish for starch, light brown for gelatin. So for the aroma of yogurt naman, only the control yogurt attained an acceptable score, and the yogurt variant with probiotics encapsulated with gelatin is the least likely acceptable. So there are significant differences among the aroma samples, the aroma as well. And a factor to consider is that yung acetaldehyde is the compound responsible para sa aroma ng yogurt. So byproduct siya ng fermentation, and the reason why hindi, and probably hindi nila na favor yung yogurt because walang acetaldehyde is supposed to be the one na responsible sa distinct aroma ng yogurt, and since wala yun, and wala yun probably because of the lower TSS of the slower post-acidification rates, which is supposed to be the one that should yield the acetaldehyde. So for the texture of yogurt variants naman, obviously there's no significant differences. And yep, no sample was deemed as acceptable. Although yung yogurt variant with probiotics encapsulated with gelatin yung most likely to be acceptable, and yung control yogurt yung least likely to be acceptable. So in relation yung to our viscosity test kanina, so no yogurt variant meet the recommended standards. However, the variants incorporated with encapsulated probiotics are more viscous or mas malapot. And it can be due to the thickening nature ng wall materials used in encapsulation. Kasi yun nga, thickeners ng sodium alginate and gelatin for instance. So, and nag-leach siya sa milk. So, for the flavor ng yogurt, so as you can see, control yogurt is the only one na acceptable, and yung variant na mayroong gelatin is the least likely acceptable. So statistically, there are significant differences, but na-comment ng panelists na medyo matabang daw yung sa variants with encapsulated probiotics, and as compared sa control sample, medyo matamis daw ang ating control. So, yogurts basically taste sour, but of course, Filipinos by nature, they would prefers sweet flavor. That's why probably TSS, control yung ating control sample yung pinaka preferable nila kasi yun yung may highest TSS content or sugar content. So medyo nag-cause din of flavors probably yung wall materials used for encapsulation. Actually, I tasted it and yeah, medyo matabang. So for the general acceptability of yogurt naman, none of the yogurt samples attained generally acceptable scores. However, yung yogurt that is most likely to be acceptable is the variant with gelatin. And control yogurt pala, sorry. And yung variant with gelatin is the least likely to be acceptable. So of course, statistically, there are significant differences, and that based on the mean scores, of course, for the other parameters, nakita naman natin na control yogurt lagi yung may highest mean scores, usually, except for texture perhaps. So that's why control yogurt ang ating most generally acceptable among the variants. So probably encapsulation does not do well in terms of the sensory parameters ng ating yogurt variants. So for the summary and conclusion, so yun nga, probiotic products are generally known para sa health benefits. However, the problem is that maraming literature reporting low bacterial counts in the commercial probiotic yogurts. So we then tap microencapsulation as a solution wherein your probiotics are immobilized in a layer of impermeable wall materials as protection. So to study the effectivity, we prepared 3 varieties. So yung dalawa with encapsulated probiotics, but yung isa has gelatin. So dalawa yung ating encapsulated variants to assess the effectivity of adding another prebiotic. So in this case, it's gelatin. So this is our findings as summarized. So for the viability, encapsulation significantly enhanced the viability, but yung addition ng gelatin has no significant effects. So variants with encapsulated probiotics have remained viable except for the control variant. So for the pH, encapsulated probiotic variants have higher pH levels due to slower post-acidification rates. The yogurt variants, however, did not meet the standard pH levels. So, ayun nga, for the TA, wala namang effects ang encapsulation and hindi din naman na-meet ang standards. So, for the TSS, yung variant with encapsulated probiotics have lower TSS, and this is in relation sa slower slower post-acidification rates. Of course, mas konting bacteria ang available para i-ferment yung lactose. So, only the variants with encapsulated probiotics have met the standards. So, for the viscosity, so encapsulation has no significant effect, and the variants with encapsulated probiotics are more viscous. So, it could be attributed sa thickening nature ng wall materials. So for the sensory parameters, encapsulation has no significant effects sa texture ng yogurt, but it has significant effects sa color, aroma, flavor, and general acceptability. So for all these parameters except texture and GA, the control yogurt is the only one na acceptable. And for the texture naman, the variant with probiotic encapsulated with gelatin attained the highest score. Yun nga, it's due to the thickening nature ng wall materials. So in all, or conclusion, the study shows that encapsulation of probiotics in yogurt significantly enhances the viability of probiotics, although the addition of a prebiotic such as gelatin had no significant effect. In addition, encapsulation has significant effects to the pH in terms of slower post-acidification rates and TSS content wherein, of course, mas lower yung levels of may encapsulation. And for the color, nagkukos ng discoloration ng encapsulation. For the aroma, of course, nare-reduce yung aroma compounds produced. And for the flavor, nare-reduce din ang sweetness na produced in relation sa lower TSS content levels. So as and general acceptability ng yogurt as well, such that mas generally acceptable ang non-encapsulated variant of yogurt. So, meanwhile, wala namang significant effects recorded sa titratable acidity, viscosity, and texture of yogurt. Okay, thank you so much.